Module 2 is concerned with looking at some of the most commonly used methods in virology. I think it's worthwhile to go through some of these methods so that you have a better understanding of how virus research is done, and also so that if you are reading something about virology, that you'll understand what they're talking about in the paper. Okay, so if you're going to work with viruses in the lab, your first problem is how are you going to grow the virus? How are you going to replicate it? Remember, viruses are obligate intracellular parasites. They have to have a host. Laboratory animals, of course, are a way to grow viruses in the lab. However, thankfully, they're not used as extensively as before, but they're still essential. Some viruses can only be grown in laboratory animals. Certain viruses will only grow in suckling mice. Primates have to be used for some types of human viruses, and mice and hamsters are used for tumor virology. If studying the pathogenesis or immunology of a virus, you have to use intact animals. This has been a big problem for AIDS researchers because to do a lot of work with HIV, you have to work with primates. And this is very expensive, but it's also very troubling for many researchers to do experimental manipulations of primates. Also, keep in mind that many of the various kinds of solutions and serums and things like that that we use in virus research do have their origin with an animal. So, for instance, if you're going to produce an antiserum, you have to use an animal for that. But there are many ways that have been developed over the years that can help you avoid having to work with a laboratory animal. And this is good, ethically, but also in terms of expense and so on. If you're going to work with laboratory animals, you have to go through all kinds of approvals. So, for instance, if you're going to work with laboratory animals here at Virginia Tech, you have to go through what we call the IACUC committee, the Institutional Use and Care Committee, and you have to submit your protocol to this committee outlining what your research is going to be, and it has to get approval from them before you can start this any kind of research involving laboratory animals. In fact, at Virginia Tech, you can't even order laboratory animals without having had your IACUC approval. And this is really good because it prevents needless experiments utilizing animals. One of the things that you have to show to IACUC is the rationale for the research. Why is this important, and why is it not just replicating other work that's already been out there? You also have to address in your IACUC protocol what you're going to do to mitigate any kind of pain or discomfort on the part of the animals. And, this is all very good. This helps to eliminate unnecessary animal suffering and ensures that the animals are treated in a humane way. But it's always better if you can avoid utilizing laboratory animals. And there are various kinds of procedures out there that have been developed over the years. One way that was developed many years ago is using embryonated eggs. And basically, with an embryonated chicken egg, if you look at the picture below, you can see that there are 4 different membranes. And each of these membranes has fluid inside of it. And different viruses grow better on different membranes. Essentially, the way the process works is you just take some alcohol and surface sterilize the eggshell. Then you drill a small hole in the shell and inject your virus into the appropriate sac. Then you just seal it up, and now you really don't have to worry about any kind of contaminations because your virus is growing in an enclosed container. This was an early method for working with viruses as a way to get away from using intact animals, but it's still used in the laboratory. We don't use it often for virus isolation anymore, but it's still used for a lot of virus vaccine production. So, for instance, the allantois produces high yields of influenza virus, and embryonated chicken eggs are still used in production of influenza vaccines. But you can see on this illustration that the chorioallantoic membrane is used for herpes simplex, for pox virus, and Rouse sarcoma virus. If you inject into the amnion, influenza and mumps viruses will grow very well. The yolk sac is ideal for herpes simplex virus, and the allantoic membrane is useful for influenza, mumps, Newcastle disease, and avian adenovirus, to name a few. So chicken eggs are very valuable in the laboratory. But what we find now is that most virus labs are going to be using some form of cell culture. The development of antibiotics was necessary before cell culture became routine in laboratories. Now it's what we normally will do when we're trying to grow viruses in the lab. There are three basic types of cell culture. First is organ culture. With organ culture, you're taking slices of organs that may function for several days or weeks. The advantage is that it maintains tissue architecture. Some respiratory viruses can only be grown in this way. Primary cultures are derived directly from excised normal animal tissue and cultured either as an explant culture or following dissociation into a cell suspension by enzyme digestion. Continuous cultures are comprised of a single cell type that can be serially propagated in culture either for a limited number of cell divisions, approximately 30, or otherwise indefinitely. Now keep in mind that although cell culture does greatly reduce the use of laboratory animals are the origin of the cells that are used in the laboratory. Let's look at primary cell lines in more detail. With the primary cell line, the tissue is surgically removed from an animal. The tissue is minced and homogenized. Collagenase is used to break down extracellular collagen and doesn't hurt intact cells. The cells can then grow as loose clumps that can be dispersed with trypsin treatment, and then individual cell types can be cultured. Primary cultures are derived directly from the excised normal animal tissue. And such cultures are initially heterogeneous, but later become dominated by fibroblasts. The preparation of primary cultures is labor-intensive, and they can be maintained in vitro only for a limited period of time. During their relatively limited lifespan, primary cells usually retain many of the differentiated characters of the cell in vivo. Most commonly used are derived from monkey kidney, human embryonic amnion, kidney or foreskin, and chicken and mouse embryos. These are used when the state of differentiation is important. Primary cell lines are also used in vaccine production because it avoids contamination with potentially oncogenic DNA from continuous cell lines. Continuous cell lines provide a uniform population of cells that can be infected synchronously for growth curve analysis or biochemical studies. These cells have been transformed. That is, they've gone through a morphological and kinetic alteration. They're usually derived from tumors or by treating a primary cell culture with a mutagenic chemical or a tumor virus. These cells are less differentiated than the cells of origin and are often abnormal in chromosome morphology and number. They grow faster, pass at low density, and are essentially immortal. They have lost the specialized morphological and biochemical attributes that were seen in vivo and have become dedifferentiated. These transformed cell lines present the advantage of almost limitless availability, but they have the disadvantage of retaining very little of the original cell's in vivo characteristics. This shows you some of the types of cultured cells that are out there, and they do vary in morphology, but you can see within an individual group of cells, they're all very similar. So on the left we see epithelial type cells, and on the right we see the long spindly fibroblast-like cells that are commonly seen. Okay, so now we know how to grow up the virus. That's step one. Now we have to be able to recognize that the virus is actually growing in culture. And there are various methods for doing this. The most commonly used are the four that are on the slide here: cytopathic effect, or CPE, heme absorption, interference, and transformation. And we'll go through each of these separately. As virus grows in a cell, changes become visible. Changes can include things like rounding up and detachment from the culture dish, cell lysis, swelling of nuclei. The cells may fuse together to form syncytia, and many other types of changes can occur. These are used in diagnostic virology to recognize virus mutants and so on. But in this slide you can see that different viruses will cause different types of visual CPE. So if we look at the far left, you can see what herpes simplex virus looks like in rabbit kidney cells. All of the panels across the bottom are what the cells look like when they're uninfected, and the panels across the top show the CPE. So you can see what herpes simplex looks like in rabbit kidney cells. In the middle you see CMV in human embryonic lung fibroblast cells, and on the right you see RSV in Hep2 cells. And notice with the RSV you're getting the formation of syncytia. In other words, infected cells are fusing together. Here's some other examples, and you can see infected cell cultures and cultures showing CPE of viruses that are commonly isolated. So in A you have uninfected A549 cells. In B you have herpes simplex virus in A549 cells, and C you have adenovirus in A549 cells. In the next row down, we have uninfected MRC5 fibroblasts, and in the middle we have CMV virus in those fibroblasts, and then at the right we have rhinovirus in those fibroblasts. The next row down would be uninfected RHMK cells, and in the middle we have enteroviruses in those cells, and at the right we have influenza virus. And at the very bottom we have uninfected Hep2 cells, and in the middle we have RSV, and then on the right is monkey virus contaminant in the RHMK cells. Morphological changes of human corneal epithelial cells infected with herpes simplex virus are also notable. In A, you have the normal human corneal epithelial cells that exhibit a cobblestone appearance. In B, you have the cytopathic effect that's seen at 8 hours post-infection, and you can see that the space between infected cells has increased. After the cells have been infected with herpes simplex virus 1 for 12 hours, you can see that what they look like at C, and then in D, the cobblestone appearance has disappeared and you have many giant multinucleated cells. So D is 24 hours after infection. But you can use cytopathic effect not only to recognize that infection has occurred, but you can get a feel for how long the infection has been taking place, how progressed it is. Now, another method for recognizing the growth of virus in culture is using hemabsorption. And this phenomenon is seen in viruses that have a hemagglutination or HA anti-receptor. Remember, we talked about influenza having an HA, but other viruses have HAs as well. And what happens is infection with the virus confers the ability of infected cells to bind or absorb red blood cells. So what's happening here is the infected cells acquire the ability to absorb or attach erythrocytes or red blood cells. And this is due to the incorporation of viral proteins into the plasma membrane of the infected cell. We see this with orthomyxoviruses, paramyxoviruses, and togaviruses. So if you look at the right, you can see what hemabsorption would look like. So you take your monolayer of cells, you place a suspension of red blood cells, uninfected now, because you've already infected your monolayer cells. You take red blood cells in a suspension and allow for an absorption period. Then you rinse them off. So only the red blood cells that have actually hemabsorbed will remain. The other cells will appear normal. And if you look over at the left, you can see what's happening. So at that very bottom insert, which is the highest magnification, you can see the top gray glob there is a red blood cell. But the line at the bottom, that is the plasma membrane of the infected cell. And you have all those virus-encoded proteins in that plasma membrane, and they are binding to the red blood cell. CPE, or cytopathic effect, is the most commonly used method to recognize virus growth in cells. And it's probably the easiest way to do it. But some viruses are non-cytopathic, so then you have to use other kinds of methods. And so, for instance, you can use the hemabsorption method that we talked about in the previous slide, but also you can use a phenomenon called viral interference. And viral interference occurs when one virus inhibits the replication of another virus. And you can use this sometimes to demonstrate the presence of a non-cytopathic virus. For example, monkey kidney cells infected with rubella virus show no CPE, but they're resistant to challenge with echovirus, which is cytopathic. So if you have cells that you think are infected with rubella virus, and then you challenge them with echovirus, if you don't see CPE, then that's your indication that you do indeed have rubella virus growing in these cells. As another example, You can look at the illustrations on this slide. This looks at two viruses, FMDV or foot and mouth disease, which does not cause plaque formation in vero cells, and then PPR virus, which does cause plaque formation in cells. So if you look at the top figure, it's just time post-infection versus virus titer, so very simple graph. And the blue line indicates the growth of PPR virus in vero cells as indicated by plaque assay. The red line, which you can see just remains flat, is FMDV, and that is flat because remember FMDV does not cause plaque formation in vero cells. So if you do a plaque assay, you're not going to see anything. And then the green line is when you had a co-infection with both, and you can see that the production of plaques is reduced when you had the co-infection. So if you look at the graph at the bottom, when you had PPRV virus alone, we just skip out to 144 hours post-infection, you can see that you have, over 3,000 plaques in your sample. Whereas if you look at FMDV alone, it's only 71 plaques. And when you do co-infection, again, the number of plaques is greatly reduced. But it depends on which one has been infected first. So if you infect with FMDV for your co-infection, the plaque formation is lower than with the PPRV. Transformation can also be used as a method for recognizing the growth of viruses in cells, in this particular case, oncogenic viruses. The morphological difference between transformed cells and normal cells is often used by scientists to detect cell transformation that are caused by these oncogenic viruses. Transformed cells are usually not inhibited by cell-to-cell contact, so the detection of anchorage-independent growth is a way to detect malignant transformation in cell cultures. In a transformation assay for mouse fibroblasts that you see in the illustrations on the slide, you can see that normal and non-transformed cells do not progress past a 4-cell stage, while the transformed cells will form a focus or colony. And you can see in the enlarged picture, basically these cells just grow up into piles or microtumors. Okay, so now the next question is, how do we assay viruses? We can grow the viruses in the lab, and we can recognize the growth of virus in the lab, but we still need to come up with methods of assaying the virus. How can we compare one strain of virus to the next, for instance? So there's a whole group of assays out there that depend on the infectivity of the virus. In other words, the virus has to be able to infect and replicate to be recognized by these types of assays. And we can further divide these types of infectivity assays into quantitative, which gives us a direct count, such as a plaque assay, a transformation assay, an infectious center assay, or a POC assay, or the quantal assays, which the result is all or none, and you will typically use an endpoint calculation method. So these are the most commonly used infectivity assays out there, and we'll go through these in more detail. The plaque assay is probably the most commonly used infectivity assay out there. It's relatively simple and precisely quantitative. It's based on the ability of a single virus particle to give rise to an area of CPE, the plaque, on an otherwise normal monolayer. There are two keys to this. First of all, you have to use a semi-solid medium. Use semi-solid because if you used a solid medium, the cells wouldn't be able to continue to grow in the monolayer. And you have to do serial dilutions. Most commonly we do tenfold dilutions, as you can see in this illustration. And the reason for doing the dilutions is so with this particular example here, we have two different virus samples. Virus 1 is across the top, and virus 2 is the bottom row. As you look across the top of each of the wells, you can see a number, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, and so on. These are the dilutions. And with plaque assays, you typically will do tenfold dilutions. So let's just look at the top row. So for this virus, when we look at 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, you don't see anything there. And that's because there was so much virus in the sample that it completely blew out the monolayer. All of the cells died. At 10 to the minus 5, now you see some purple spots. Okay, those purple spots are from the crystal violet that we use to stain. So we typically use this kind of system, this sort of stain in my lab. And what we do is we have our monolayer growing, we put the virus on, then we'll put our semi-solid overlay. In our case, we use methylcellulose. And you want it to be a semi-solid, but not too hard, because you're going to have to remove it eventually. So the proper consistency really for the methyl cell that you would use in an assay like this would be similar to what you would have in a Jell-O shot. Not that I've ever had a Jell-O shot. So with the 10 to the minus 5, you can see that you still have some live cells, but most of the monolayer has been wiped out. When you go on to 10 to the minus 6, you can see that you can clearly see individual plaques. Those are the white spots. Remember, the purple is from the crystal violet stain, and that indicates the cells. However, there's still places where individual plaques have coalesced together, so you can't get a good count. So I would consider 10 to the minus 6 for this particular virus sample to be at the 2 numeristic counter, TNTC level. But if we move on to 10 to the minus 7, we can clearly see individual plaques. So like Goldilocks, this is the perfect one. 10 to the minus 7 is the well that I would choose to pick to do the count. But keep in mind that when you're working your results for a plaque assay, you have the number of plaques, you also have your dilution factor, but you also have your volume. You don't just say, well, we had 100 plaques. You had 100 plaques per what? Is it per ml, per 0.2 ml, whatever. So you have to have a unit of volume. Now, if we go down to the second virus in the bottom row, you can see that at 10 to the minus 3 is where you would do your count. And this tells you that there's far less virus in that second virus sample than what we saw in the first. And again, you would count your plaques, take into account the dilution factor, and report it as number of plaques per volume. And this gives you a really good way to compare individual virus samples. Another advantage that you have in working with plaques is, remember, Each one of those plaques arose from a single virus particle. The semi-solid medium prevented the virus from spreading anywhere except to cells that were directly adjacent to the originally infected cell. That's why we use the semi-solid medium, so that the virus can't be released into a liquid overlay and then infect all of the cells in your monolayer. So that gives you the advantage that each plaque is essentially a clone, a genetically pure line. So you can go in and take a sample of an individual plaque, and we call that plaque purification. You've essentially cloned your virus by doing that. We also have transformation assays. This is the kind of assay you would use with an oncogenic virus. Remember, with transformation, the infected cells change their behavior so that they take on the properties of malignant cells. This is very similar to a plaque assay in technique and function. The only difference here is that you're counting foci or microtumors rather than plaques, but the basic methodology would otherwise be the same. Infectious center assays are a modification of the plaque assay. In this case, you can determine the number of cells in a culture that are infected with virus. So compare this to a plaque assay. A plaque assay uses a virus suspension. The ICA uses infected cells. You plate dilutions of infected cells on a monolayer susceptible cells, and then the number of plaques is an indicator of how many cells are in the original sample that were infected. In the figure, we're looking at 2 samples of equine herpes virus. The virus infects and replicates in the peripheral blood mononuclear cells of the horse. The question we're asking is what percentage of blood cells are infected. So we take a sample of blood from the horse and do a dilution series. Then we add the blood suspension to a monolayer of cells. You will get a plaque forming where there were infected cells. Again, the number of plaques, the dilution factor, and the volume are important. In this figure, the top virus, the wild type, had a higher titer, or a higher percentage of infected cells, than the virus sample at the bottom. For viruses that grow in embryonated chicken eggs, we can use the POC assay. In this case, we're titering the virus on the chorioallantoic membrane, or CAM, of the chicken egg. So we just use the typical methods that we discussed earlier in this lecture, and viruses will form focal lesions on the membrane. And you can see these red blotches. Those are the pox. So you would introduce the virus into the chicken egg, allow time for the pox to grow. Then you can introduce a stain, break open the egg, and actually count the individual pox. This has mostly been replaced by cell culture techniques for obvious reasons, but it's still used in vaccine production, and it's a specialized assay for different types of pox viruses. So all of the tests that we've talked about so far have been quantitative. There's something that you can count to compare, whether it's plaques or pox or infectious centers, microtumors, and so on. But we have a second group of tests that are quantal assays. With these types of tests, the result is all or none, and you have to use a different kind of approach then. So for instance, think of an LD50, a lethal dose 50%. That would be a quantal assay. And it's considered all or none because either the animal dies or it doesn't. You don't have any in-betweens. And so because for each individual unit you have a yes or a no kind of result, you have to have multiple replicates for each dilution. And you have to put those into your assay system. And then you can calculate out an endpoint using statistical analyses. You can use, for instance, the Reed-Munch method. That's a very easy one to use. But examples of these kinds of tests would be TCID50 or tissue culture infectious dose 50%, EID50 or egg infectious dose 50%, or LD50 or lethal dose 50%. Now, oftentimes you'll see 50% as the endpoint, but it can be higher. You'll often see 90%, an LD 90 or 95, but you will never see 100%. And if you stop and think about it, that makes sense because it's very difficult to try to calculate the minimum dose of something that will cause 100% of the replicates to die. Because remember, any dose above that will also cause 100%. The key to calculating an endpoint, whether it's 50% or 90%, 95%, you have to bracket the endpoint. So for instance, if you're calculating an LD50, you would have to use doses that cause mortality rates below 50%, and you would have to use doses that cause mortality above 50%. But the easiest way for us to understand this is to look at an actual calculation of one of these endpoints. So in this slide, we're going to calculate the TCID50 or tissue culture infectious dose 50%. If you look at the picture at the bottom, that's a 96 well plate. And what we have done is put in cells and allowed them to grow in a monolayer in each of the wells. Then we're going to introduce our virus. So for each well, because we're using a liquid overlay, so it's not a plaque assay, we're using a liquid overlay, when the virus starts to replicate in cells, it can spread to all of the cells within the well and blow out the entire monolayer. So for any well, we will either have no growth, uninfected, or will have infected. And so if you look at how this plate is set up, you see 10 to the minus 1, 2, 3, 4. And you'll see that there are multiple wells going across at 10 to the minus 1. Those are replicates. Each one of those wells received a 10 to the minus 1 dilution dose of the virus. Then you go down to minus 2, and again, across the row, you have a series of replicates. Now the other thing with this is the dark wells indicate infected, the ones that are dotted indicate uninfected, and then the ones that are blank, those just were not used in the assay. So what we're going to have to do now is count up the numbers of infected wells. And it's just very simple. You go through, and the calculation is up at the top of the slide. So you need to count up, you figure out which dilution was next above 50%, you count up the number of wells, and you figure out which was the dilution next below. So let's look at the next slide, and we'll actually do the count. Okay, so we still have the formula at the top. This is the same plate, but you can see if you go across at 10 to the minus 1, 100% of the wells were blown out. 10 to the minus 2, 100%. 10 to the minus 3, 100%. 10 to the minus 4, we're at a lower dilution of virus. Now only 60% of the wells were blown out. At 10 to the minus 5, only 20%. So this particular spread of dilutions worked for us because remember I said you have to bracket the endpoint that you're looking for. So in order to be able to do this calculation, we would have to bracket it 50, and we did. So here you take the number of wells infected at the dilution next above 50%, that's 60, and you subtract 50 from that. And then for your denominator, it's the number of wells at the dilution next above 50%, so 60, and then the number of wells infected at the dilution next below 50%, and that would be 20. So that gives you 10 over 40, or 0.25. So our titer, you see in the bottom right corner what our titer is. So 10 to the minus 4, that dilution got us to 60%. So our final titer is going to be somewhere between minus 4 and minus 5. And so we take that 0.25 and essentially just tack it on to the 4. So we have 10 to the 4.25. And notice how this result is written out. It's 10 to the 4.25 TCID 50. So you have to write down what kind of a unit it is. So it's tissue culture infectious dose 50%. And then you see it's per 50 microliters. Just like all of these other kinds of assays we've talked about, you always have to put your units in, because we could have added 20 microliters per well, and that would give us a completely different dose, different result. So this is how you would do the calculation. And here's another example. With this, we're calculating LD50, and we're using mice, and you can see over at the left the different dilutions of virus, -1, -2, and so on. Now, for each one of these dilutions, there were 10 mice. So 10 mice were given an injection with the dilution at -1, and 10 other mice were given -2, and so on. And then, after the appropriate time period, you count up how many died and how many survived. And see, that's why I said these are quantile assays, they're all or none, because you don't determine which mouse might have gotten sick or so on. You look at live or dead, and then you get your cumulative totals and figure out your percent mortalities, which you see at the side here. And then you just do, at the bottom, there's just another Reed-Munch calculation. And you can see that in this case, the titer of the virus was 10 to the 6.23 LD50 per microliter. Here's a comparison of the toxicities of a number of different compounds. And the toxicities are indicated by the LD50. And in this case, they've adjusted it for the body weight of the organism. In our previous example with the mice, when you do a study with mice, you get mice all of the same age, so that their body weights are pretty darn similar, so you don't usually have to worry about it. But you can see that highly toxic chemicals have a small number first. 1 gram per kilogram is much less toxic than 0.1 micrograms per kilogram. So that's something that you have to be aware of, that your units can differ. And it means that you can scale up and down. And of course, these kinds of considerations become important too, because obviously a mouse dies from much less poison than an elephant. Likewise, you could outdrink a baby with tequila shots. So that's why when you're looking at comparisons of multiple compounds like this, you have to include the weight of the organism. But when you adjust for weight, oftentimes you'll find that you will have a proportional amount of toxin per body mass to yield similar levels of mortality. Okay, then we have physical assays. Physical assays don't measure infectivity. So the virus doesn't have to be live and infectious. The most common types of physical assays that we do involve electron microscopy or hemagglutination. Direct particle count is the method that we would use if we're going to use electron microscopy. And what you do is you mix your virus sample. So of course you don't know how many virus particles are in your sample of virus. That's what you want to find out. So you'll mix your sample of virus with the known concentration of latex beads. So you know exactly how many beads per ml are in your sample. Then you do a count of both, and you can calculate out the concentration, because if you have 10 beads and you know what the concentration of your sample was, then you can calculate out what your concentration of virus is. You need to do this if you're working with electron microscopy, because you can't really control exactly for the area that you're looking at under the scope. So you need to work with concentrations. This is effective, but it's time consuming. And of course, you can't distinguish between infectious and non-infectious particles. And here's an actual electron micrograph showing you how this would be done. So in this case, the on this particular field in the SEM, you have 15 latex beads, and those are indicated here by the yellow arrows, and 14 vaccinia particles. They look a little bit more brick-shaped. So from this you can then do a calculation, because if you know what your concentration of latex beads was, and then you count up your number of virus particles, then you can make that calculation. However, we find that as with most things, they've been able to come up with automated systems for doing it. So now there's a virus counter device. This instrument detects and measures virus particles in a fluid stream. And it's a pretty cool device. And if you look at the graph at the bottom, they used their virus counter to calculate the number of virus particles in their samples, and they compared that using electron microscopy, so that's the gray bar, and then they did a TCID50, or tissue culture dose, tissue culture infectious dose 50%, to look at infective particles. Because remember, the TCID50, it's a quantile assay, but it's based on infectivity of the particle. So that's going to count up only the infectious particles. So here they took samples of influenza virus, cytomegalovirus, respiratory syncytial virus, and rubella virus. And they compared across these three different platforms. And what they found was that the virus counter results were equivalent to what they found using TEM, using the electron microscopy. But the titers determined by TCID50 were 2 to almost 4 orders of magnitude lower. These results highlight the relative abundance of non-infective particles across multiple virus types. Okay, then we have a hemagglutination assay. With the hemagglutination assay, you can use this for viruses with surface proteins that can bind RBCs. So adenoviruses, orthomyxoviruses, paramyxoviruses all show this. And we've already talked about hemagglutination, or I should say hemabsorption. So it's a similar kind of thing. With hemabsorption, the red blood cells were binding to the surface of an infected cell, but it was based on the ability of proteins produced by the virus to bind to RBCs. So in this case, rather than working with infected cells, we're working with suspensions of virus. And the virion will attach to multiple RBCs and link them, forming a lattice. So the key to this assay is that you have to use a round-bottomed well. The non-aggregated blood cells will settle out just by gravity and roll to the bottom of the well and form a button. So you can't use a flat-bottomed 96 well plate for this. You would use a flat-bottomed 96 well plate for a TCID50, but for a hemagglutination assay, you need a round-bottomed well, because these non-aggregated blood cells have to be able to settle out. The aggregates that are formed by linking between red blood cells and virions will also settle to the bottom, but they'll form a film. And you can see that. So in this picture at the bottom right, you have the red dots. That would indicate that you didn't have hemagglutination. The non-aggregated blood cells settled out and formed the button. That's what we call that dot. It's a button. But in some of the other wells, you see it looks like a pink smear. And that's where you have your lattice. That means that you did have the attachment. You did have absorption. Now, there are advantages and disadvantages to this kind of a test. It's a very rapid test. You can get your results in 30 minutes. But it's not sensitive to small amounts of virus. And here we'll just look at this some more. So you have red blood cells and your virus. Those are your components. Across the top at A, you can see if you don't have virus, you don't get an interaction between the blood cells. They're going to settle out and form the button. Across in B, if you put in the virus will bind up with the red blood cells, and you get hemagglutination. So instead of the button forming, you get that pink kind of a smear. And we can get a titer using this kind of an assay. So here you can see on this particular plate, they use 7 different samples of influenza virus, so 1 through 7 over across the top. And they did serial dilutions and mixed it with red blood cells. The wells in the bottom row contain no virus. That was your control, because you always have to have a control with these. Now, you'll notice that as the virus becomes more dilute and the dilution is across the top, and the numbers are different here because with the hemagglutination assay, remember, it's not sensitive to low amounts of virus. So you don't use tenfold dilution. Generally, you use like a twofold or fourfold dilution. So you have a different dilution series, but you can see that it's been serially diluted across the top. And what you would do is go across each row and look for the well where you no longer see complete hemagglutination activity. And what we have here then on the right side is the number, the dilution, at which point you no longer saw complete hemagglutination activity. So 1024 for virus 1, 64 for virus 2, and so on. So the question is, which virus sample had the highest concentration of virus? And actually you have two that are tied, number 1 and number 7. They both have a titer of 1024, and so with what that means is that those two viruses had to be diluted out further than any of the other viruses. So that means that there was more virus in the sample. So you can see here from this slide, you have different efficiencies for different kinds of assays. They're not all created equal. So on this slide, we're showing the kinds of results you would get using different types of assays for influenza virus. So with the direct particle count, there's no surprise you're going to get a higher titer with this, because remember, this is not going to count the non-infectious particles. The Quantal assay in eggs, when you're working with influenza, is a very sensitive assay, so you can see it's 10 to the 9th egg, ID 50 units. Plaque assay, 10 to the 8th PFU, and hemagglutination assay, it's 10 to the 3. So this shows you, number one, if you're trying to compare results from one lab to another, one paper to another, and so on, you have to take into account the kinds of assays that were used. You can't make a direct comparison between an egg assay and a plaque assay. Also, you might think to yourself, well, why would you use something like hemagglutination? You might think, well, the plaque assay is a really good assay. It's easier than eggs, and you get a nice high titer. I'm going to do plaque assays. Well, that will work for you, and that's true, but remember, plaque assays require cell culture, so you have to be set up with the proper CO2 incubators to do cell culture. You have to have the technology, and it's going to take you several days and involve several steps. Remember, you put on the overlay after the absorption period, then you have to go back and stain and so on. Whereas with the hemagglutination assay, it's 10 to the 3 HA units because we said it's not sensitive to low amounts of virus. But it'll give you your result in 30 minutes. It only requires a suspension of red blood cells, which you can buy commercially very cheaply. And it doesn't require the skill and equipment that's required with the plaque assay. So a hemagglutination assay can be a very excellent screening system to use. So that's the big take-home lesson with all of these assays. There's all kinds of different assays out there, and you choose according to your budget, your expectations, what you're trying to accomplish here. So then we can move on to serological and immunological methods. Often it's difficult to identify a virus in relation to the disease observed, or when conducting a retrospective study of a population to determine exposure to a virus, or when measuring the response of an individual to a vaccine. In these cases, indirect measures are needed, such as measuring antibody response to the virus of interest. Several methods exist for this purpose. We will consider some of these, but the principles of these assays are fundamentally the same. They depend upon antibody-antigen interactions and consist of a known virus or viral protein, a patient sample, and an indicator. If antibodies are present in the sample, often a patient serum, they will bind to the virus. If no antibodies are present, no binding will occur. The indicator is observed to determine whether the sample is positive or negative for antibodies. So on this slide, you can see some of the most commonly used serological or immunological methods out there, and we'll go through some of those. So first we want to look at neutralization tests. In the virus neutralization test, the sample of interest is incubated with the target virus and changes in cell culture observed. So we're looking for changes in cytopathic effect. If the sample contains antibodies, it will prevent the virus from growing in the cell culture, and no CPE will be observed. If no antibodies are present in the sample, the virus will grow and CPE will be observed. Specific types of antibodies can bind to surface proteins of the virus and block an early event of the virus recognition or effective internalization of virus. Dilutions of serum will be mixed with a constant dose of virus. You're going to incubate, usually for about 60 minutes, because you want to allow time for the antibody and the virus to bind. And then you assay for residual infectivity. What that means is that you, as your control, you have to titer your virus sample without the antibody. So you have to know how much virus you have. Then you incubate with the antibody, and some or all of the virus particles will be neutralized by your antibodies, and then you do a test to see if there's any infectivity left, or the residual infectivity. It's very common to look for plaque reductions. The endpoint would be recorded as the highest dilution of antiserum that inhibits virus. This is a very sensitive and specific test. It's directed against surface antigens involved with absorption to the host cell. Now, these antigens are under the most selective pressure, so they're the most specific to viral type and strain. Usually there's little cross-reactivity to related viruses when you're using a neutralization test. And also, NT antibodies tend to be of very long duration. They're IgG antibodies. So you can go back, many months and sometimes even years after somebody's been infected and still get neutralizing antibodies from a serum sample in those patients. In this slide, you can see an example of a plaque reduction neutralization test, or PRNT. This is very commonly done. Plaque reduction neutralization tests are extremely sensitive and specific. So in this particular example, you're looking for antibodies to coy herpes virus, or KHV. KHV is a highly contagious virus that causes significant morbidity and mortality in koi fish. This is significant because some types of koi fish are quite expensive. The tissue cultures were of koi fin cells. You use diluted serums from blood samples from fish, so you're wanting to see if the fish have antibodies or not. And then you used KHV samples to incubate. And you incubated at permissive temperatures and ran your plaque assay. Ultimately, you fixed and stained the plates and counted up the plaques. In samples where antibody to the KHV was present, the virus did not grow or was severely inhibited. So if you look at the bottom right, you can see that there are no plaques at all in that well. That's an indication of a high level of antibodies. If you look at the well on the lower left, on the other hand, you still do see some plaques, but greatly reduced compared to the samples that were negative. So that's what I meant by looking for residual infectivity. When you incubate the antibody and the virus together, depending on the titer of virus and also depending on the titer of antibody, you may not get complete removal of virus infectivity. You may not get complete neutralization, but you can still get a significant reduction, and that's what we see in the bottom left. Immunodiffusion is another kind of assay that can be done. It's often referred to as the agar gel precipitin test, and it involves the diffusion of virus and antibody through an agar, which will form a line of identity where the antigen-antibody complexes form. So you just use octoloni plates with your agar. You cut holes in the agar, and into these wells that you've just made, you put your reactants. Your reactants, some of the wells will contain the antigen of interest, some will contain the antibodies. And because it's agar, these liquid reactants will be able to diffuse through the agar, and eventually, when they come into contact with each other, if they're a match, you get a line of precipitation. This test is very simple. It runs very quickly, doesn't require a lot of fancy equipment or anything like that, and you have the advantage too that you can test several antibodies or antigens at once, so you can have multiple wells. But there's no quantification here, and it requires a high concentration of reactants. So here you can see some results in these two different plates. Antigen was added to the center well, so that was your test reactant. And then in wells 1 and 4, those are your positive controls, so you know you have antibody that matches the antigen in the center well there, so you have to have a test. With samples 5 and 6, those are patient seras that happen to be negative. You don't see any line of precipitation. In patients 2 and 3, those are positive tests. You can see the lines of precipitation there. Another test is complement fixation. Complement is a protein component in serum that affects cell membranes. It causes cell membranes to lyse. Its action is triggered by antigen-antibody complexes to initiate a cascade of proteolytic cleavage and protein binding reactions. It's an important component of our immune response. However, there's a lack of specificity of complement, and that allows it to react with almost any antigen-antibody complex. Therefore, in this test procedure, Unless complement is fixed by the particular antigen and antibody system in question, it will remain free to react as an indicator, resulting in a negative test. If, on the other hand, complement is fixed by the antigen and antibody system under study, it is not free to react in the indicator system and results in a positive test. The indicator system used in this particular example, it would be sheep red blood cells, RBCs. In a positive or reactive test, the complement is bound to an antigen-antibody complex, which is what you see in the left row, or left column, and it's not free to interact with the sensitized red blood cells, so they remain unlysed and settle to the bottom of the well to form a button. On the right side, though, you have a negative or non-reactive test. The complement remains free since there's no antigen-antibody complex for it to bind to, and it interacts with the sensitized RBCs, causing them to lyse. So it's an easy system to use. It doesn't have to, it doesn't require a lot of expensive equipment. You get your results very quickly. But there are disadvantages. For instance, very precise controls are necessary, and even things like a high lipid content in the serum can affect the results. So, for instance, you'd need to make sure that the patient had been fasting before the blood sample had been taken. If they'd stopped at Hardee's drive-thru on the way over, it would throw the test completely off. It's also not very specific, but it's quite useful as a preliminary screening. You can use it to identify the group to which the infecting virus belongs, and then you can follow up with more sensitive tests like the plaque reduction neutralization test to get types. So if you have a large number of samples, you can quickly screen them using a test like complement fixation, and then just use the plaque reduction neutralization test or other kind of sensitive test to confirm your results. And run those tests, of course, just on the positives. Then we have hemagglutination inhibition. Now, we've talked about hemagglutination already. Remember, certain viruses have a protein on their surface that interacts with red blood cells and is able to attach to them. This property is called hemagglutination, and the surface protein of the virus is the hemagglutinin. The inhibition or blocking of this activity is the basis of the hemagglutination inhibition test. So it's kind of the flip side of the hemagglutination test that we just talked about. The most well-known virus with this hemagglutination property is influenza virus. Like the virus neutralization test, the patient's serum is going to be incubated with the virus of interest, but instead of growing the virus in cells, red blood cells are added to the serum virus mix. If antibodies are present, the hemagglutination activity will be blocked. If no antibodies are present, the virus will agglutinate or bind together. In this case, the red blood cells are the indicator. So you mix dilutions of serum with a constant amount of virus, and the titer is the highest dilution of serum that inhibits hemagglutination. Let's look at an example. So here you can see the kinds of results that you would get. Across the top, you have your red blood cells. Your sample has no virus, so you get the button formation, no reaction. If you add just virus and red blood cells, you get the lattice formation, you get that pink smear. That's hemagglutination. Remember, we looked at that already. But if you have virus and antibody, the antibody binds with the virus, and that prevents the hemagglutination from occurring. And then you get the button again. And that's why they call it hemagglutination inhibition. You add antibody to bind up the virus so that the virus can't bind up the red blood cells, so you don't get hemagglutination. And at the bottom you can see the kinds of results that you would get in wells. So as you dilute your antibody sample out, you go from having buttons, meaning that you have inhibition, to the point where you've diluted out the antibody and now you have hemagglutination again. And here's an example of how you would calculate a titer. Again, you've got your virus control at the bottom. As the virus becomes more dilute, the hemagglutination disappears. So for this virus, we expect hemagglutination to occur. As you dilute your virus, hemagglutination disappears. But we're using a constant concentration of virus here. What we're diluting instead would be your serum. And what you're doing then is diluting your, or I should say you're incubating your dilutions of serum with a constant amount of virus. If the serum antibodies bind virus, no hemagglutination occurs and the button forms. So here you can see your titers at the side. The titer for serum 1 sample was less than 10. So, we can't say that it was a negative sample because there's not sufficient sensitivity to the test. But certainly less than 10. With serum 2, we'd call the titer 40. Serum 3, 640. And serum 4, 2560. So we've had to dilute out the serum further. And again, across the top, those are dilutions of your sample. And you can see we were using twofold dilutions here. So again, you use different dilution series depending on the type of test that you're running. This is a sensitive, cheap, and rapid kind of test, and it works well for any virus that causes hemagglutination. But here, so which serum sample has the highest concentration of antibodies? Of course, it's going to be serum sample 4 because you had to dilute it out further than any of the other samples to get to the your endpoint. And again, this is very sensitive, cheap, and rapid. It's very useful as a screening test. As an example of how this test can be used, when I was a graduate student at the University of Notre Dame, one summer I worked on a project for the Indiana Department of Health. They were going around collecting serum samples, blood samples, from wild birds all over the state. And these samples were brought back to the lab at Notre Dame, and we were going to test them to see if they had antibodies to some of the arthropod-borne viruses that use wild birds as a reservoir. So we were getting as many as 500 samples a week. And there were just 3 of us working. 2 of us were part-time because, we were grad students and had to do research and stuff like that. And 1 full-time person. And with just that level of staffing, we were able to go through the 500 samples a week and still be able to show up for the 4 o'clock happy hour on Fridays. So it was a very rapid system, and it was very inexpensive as well, because you just needed your solution of red blood cells and things. You didn't have to use cell culture and any other kinds of methods that tend to add a lot of expense and require more expensive equipment in order to run it. Another test is radioimmunoassay. with an RIA system. As the name indicates, it's an immunological assay to analyze any antigen or antibody in the patient's serum. You can use it to diagnose a disease. It's one of the most sensitive and specific methods of immune assays available. And it involves the competitive binding of radio-labeled antigen and unlabeled antigen to a high-affinity antibody. If a lot of antigen is in the experimental sample, it competes with labeled antigen for binding sites on the antibody, and then you'll see very little radioactivity bound. If there's a high amount of bound radioactivity, then there is little antigen in the sample, and then you just generate a standard binding curve. This used to be used very frequently for detecting the presence of hepatitis B surface antigen in donated blood. We have more sensitive and easier to run kinds of tests that are used now, but this was very widely used by blood bank systems for many, many years. The IFA is a rapid method of virus titration that allows the quantification of virus in cell lines that do not support plaque formation or do not exhibit detectable CPE, such as hepatitis A virus. You can also use IFA to see which cells in a monolayer are infected. In contrast to other infectivity-based assays, IFA is not dependent on the ability of induction of cell death, but it utilizes an antibody-based staining method to detect virally infected cells. Fluorochrome conjugated antibodies react with appropriate antigens and are detected by an immunofluorescence. Fluorescein is the most common conjugate, resulting in a bright apple green color by fluorescent microscopy. Direct immunofluorescence involves direct application of fluorescinated antibody to the specimen. Indirect immunofluorescence entails mixing non-conjugated antibody with the specimen, followed by application of a fluorescinated antibody targeting the initial antibody. So, for instance, you might use a mouse anti-human and then a goat anti-mouse to form what they call a sandwich, as you can see in this figure. The indirect technique is usually more sensitive than the direct technique. In this illustration here, you see IFA of viro cells infected with the Chilean hantavirus isolate. On the left is IFA with seropositive human sera from a hantavirus patient. The arrows show infected viro cells expressing hantavirus antigens. On the right, you see IFA with seronegative human serum from an uninfected control. The arrow shows the negative IFA of viro cells infected with the virus. ELISAs, or EIAs, they're the same thing basically, are very popular techniques because of their ease of use and low cost. The test consists of plastic wells that have been coated either with the antigen or virus of interest or a protein specific to the antigen or virus of interest. The unknown sample, the serum, is allowed to bind to the coated well. An antibody labeled with an enzyme is applied, and an indicator is added, and then a color change is observed. The presence of color indicates the presence of antibodies, and the absence of color indicates the absence of antibodies. This can be used to detect either antigens or antibodies. In order to get specific results, though, you have to use a reader. This just shows you how you can run a test looking for antigen or run one looking for antibody. So on the left, with the antigen capture test, you have your well that's been treated so that there's a specific antibody attached to the bottom of the well. You add your test specimen, which may contain the antigen. If it contains the antigen, it binds to the antibody. And now, if you've ever done these tests, you know that you put it through washers. So you're running this in a 96 well plate. The washers will come down and wash out each of the wells. In the wells where antigen has bound to the antibody, that antigen will not wash out. If the antigen is not a match to that antibody, it won't bind and it will wash out. Then you follow up in step 3 with your labeled enzyme, which will match to the bound antigen. You do another wash. Then you add your substrate, and if the enzyme has been linked, it will trigger a color change in your substrate, and then you put it on a plate reader. For antibody capture, in this case, instead of starting out with bound antibody, you start out with bound antigen. Then you add your specimen, which may or may not have the antibody. If it's a match, it will bind to the antigen. And again, remember, in between each of these steps, you go through washes. So any reagents that have not bound to the original antigen will be washed out. So then you add your enzyme labeled antibody and allow that to bind. And then your last step again is adding your chromogenic substrate. And if your labeled antibody with the enzyme was bound in place, you'll get a color change. And again, you put it through a plate reader. Here's an example of a result of one of these tests. This test was done on patients looking for HIV infection. The ELISA tests, EIA tests, are very commonly used to detect HIV infections. And you can see the colors here. Again, to be very accurate, you would have to use a plate reader, but we can see these by eye. So the dark green is your positive well over on the left. Then you have the light green is your negative. And then you have 3 patients, A, B, and C, and then you have your assay control. Now, when you look at patient C is a very strong positive. Patient A is a negative. But then you look at patient B is equivocal. That result is technically below the cutoff value, but it's very close. So that patient would probably be asked to return after a period of time, because what may have happened is that they were being tested very early in their infection and they hadn't developed high enough antibody level to be detected. Usually, these positive tests are confirmed by Western blotting. And with a Western blot, again, you're looking at specific virus proteins and a complex of antigens. And you're using electrophoresis, and you're going to probe with antibodies. So it depends on the reaction of antibody with the protein, but it's in a different format. In this case, it's been immobilized on a thin membrane. So first you take your proteins and you fractionate them just using PAGE, polyacrylamide gel electrophoresis, and then you blot it or transfer it to a thin synthetic membrane that has a high affinity for proteins. The proteins will then be detected by immunostaining. And this is also used for HIV. So on this particular, in this particular example, if we look down lane 1, GP 160 is a viral envelope precursor. GP 120 is the viral envelope protein. GP 24 is viral core protein. And P31 is the reverse transcriptase. So this is your positive control here, and this shows you the kinds of proteins that you would see when you have an HIV infection. With lane A and B and C, you have patient serums. Patient A is negative. You don't see any positives for the virus. Patient C is very clearly positive. And again, patient B is indeterminate. So another test would be ordered in this case.